For our next panel, we will hear from three experts on the structural chemistry and pharmacological effects of fentanyl and fentanyl analogues. Our panelists are Dr. Michael Van Lin, Dr. Srihari Tella, and Dr. Barry Logan. Dr. Van Lin is a drug science specialist chemist in the Drug and Chemical Evaluation Section, Diversion Control Division, Drug Enforcement Administration. His professional responsibilities include collecting data and information on newly emergent psychoactive substances and chemical diversion trends, evaluating available information and drafting scheduling actions, providing scientific insight for domestic and international drug control policies, and providing expert testimony in controlled substance analog prosecutions. Dr. Van Lin earned his PhD in organic chemistry from the University of Wisconsin-Milwaukee and conducted postdoctoral research at Drexel University College of Medicine. He earned his undergraduate degree in chemistry and biology from the University of Wisconsin-Stevens Point. Dr. Tella has been a unit chief of the Drug and Chemical Control Unit of the U.S. Drug Enforcement Administration since 2009. His current responsibilities include managing a group of scientific staff involved in reviewing drugs and other substances with abuse potential for control under the Federal Controlled Substances Act. Dr. Tella's unit routinely provides support to federal prosecutors by serving as scientific experts in federal drug cases pursued under the Controlled Substances Act. Dr. Tella earned his PhD in pharmacology from the All India Institute of Medical Sciences, New Delhi. He received his postdoctoral training at National Institute on Drug Abuse, National Institutes of Health in Baltimore, and following postdoctoral training, he served as a faculty member in the Department of Pharmacology of Georgetown University School of Medicine. Dr. Logan is a vice president of forensic science initiatives at NSM Labs, a leading United States provider of esoteric toxicological testing services, specializing in new drug detection and forensic analysis for the prosecution and defense in criminal justice and death investigation agencies. He has over 90 publications in toxicology and analytical chemistry, including treatises on the post-mortem redistribution of drugs and the toxicology and chemistry of novel psychoactive substances. Dr. Logan is board certified by the American Board of Forensic Toxicologists. He graduated from the University of Glasgow with degrees in chemistry and forensic toxicology. Dr. Logan did his postdoctoral training in forensic toxicology at the University of Tennessee Center for Health Sciences. Dr. Van Lin.
Good morning, Chair Pryor, distinguished members of the United States Sentencing Commission. Thank you for the opportunity to briefly discuss chemical structures of both fentanyl and its analogs. Fentanyl belongs to the 4-annalidopyridine structural class, which is a group of substances that have been well studied for their potential analgesic effects. Fentanyl analogs have chemical structures that are similar to that of fentanyl, but with small chemical structural modifications. The synthesis of fentanyl and fentanyl analogs requires some familiarity with synthetic organic chemistry. However, the design of new fentanyl analogs is quite straightforward. With substitution of one or more of the chemicals used in the synthetic process, a new fentanyl analog can be produced. The scientific literature describes the diversity of structural modifications that have been made to this chemical structure of fentanyl to create structure-activity relationships, often accompanied by pharmacological data and detailed instructions for their synthesis. Some fentanyl analogs have emerged on the illicit market, were previously reported in the scientific and patent literature. The ease of creating new fentanyl analogs is attractive to medicinal chemists researching the structural class. Unfortunately, the same can be said for the clandestine chemists. They can utilize the same scientific knowledge and continually produce fentanyl analogs with the intent to circumvent laws. The recent dramatic increase in trafficking and abuse of fentanyl and fentanyl analogs are tightly intertwined with the opioid abuse in the United States, which is already at alarming levels. This topic needs little introduction. However, I would like to mention a common occurrence that I've observed while working on 6 emergency control actions to control 9 fentanyl analogs in Schedule 1. The DEA collects reports on fatal overdoses involving fentanyl analogs on an ad hoc basis from state and local medical examiner and toxicology labs, as well as coroner's offices. One case describes the fatal overdose of a 20-year-old male from Maryland. The victim was found unresponsive in his bedroom, kneeling on the floor and leaning over a bed. Upon moving this individual, a syringe was discovered in his hand. Toxicology revealed that the decedent died of an overdose of heroin and acryl fentanyl, which is a fentanyl analog that is similar in chemical structure to that of fentanyl. Many of the fentanyl analogs that have been temporarily controlled have been involved in similar cases. Other fatal overdose cases have occurred with many of these fentanyl analogs, where a needle was still found in the decedent's arm. Others have occurred at drug treatment facilities, and others have involved multiple victims at the same location. The DEA is closely tracking fentanyl analogs as they emerge on the illicit market, both internationally and domestically. Unfortunately, in many cases, we are notified of a new fentanyl analog when there are deaths and the substances are identified in postmortem toxicology. When we learn of a new substance, we immediately begin collecting data and information on that specific fentanyl analog. This information is evaluated and utilized to support a potential scheduling action. Since 2015, the DEA has completed 6 temporary scheduling actions to temporarily control 9 fentanyl analogs in Schedule 1 of the Controlled Substances Act. We are also made public notice of intent to control another fentanyl analog in Schedule 1, a substance known as cyclopropyl fentanyl, which you've heard from Major Clone in the previous panel. Cyclopropyl fentanyl, like other fentanyl analogs, has a chemical structure that is closely related to fentanyl, but has not been previously reported in the scientific literature. We first became aware of this substance in early June of 2017 when it was identified in white powder submitted to a local drug forensic chemistry laboratory. The following week, the media reported on a group of more than 40 overdoses in the state of Georgia involving counterfeit Percocet tablets. Upon analysis, cyclopropyl fentanyl was identified in combination with another substance known as U47700, which is another synthetic opioid. The presentation of cyclopropyl fentanyl in powder form and disguised as an opioid pharmaceutical product show that fentanyl analogs are introduced to target the opioid-dependent population. In summary, the fentanyl analogs have been identified in drug evidence and postmortem toxicology show that traffickers are utilizing the scientific literature to introduce already known fentanyl analogs, as well as using the information from structure-activity relationships to design new fentanyl analogs for introduction into the illicit drug market. Responding to the introduction of new fentanyl analogs in the illicit drug market remains a priority for the DEA, and we look forward to working with the Commission to address these substances. Thank you for the opportunity to discuss this important issue, and I'll be happy to answer any questions you may have.
Thank you, Dr. Van Lin. Dr. Tella?
Good morning, Judge Pryor and distinguished members of the Sentencing Commission. I thank the Commission for providing this opportunity to discuss pharmacology of fentanyl and related substances. Pharmacological and toxic effects of fentanyl and its mechanism of action are in general similar to heroin and various commonly used opioid analgesics such as morphine, hydrocodone, and oxycodone, etc. However, there are 2 main differences between fentanyl and other commonly used opioid analgesics. The first difference relates to the potency. Fentanyl is more potent than commonly used opioid analgesics and heroin in producing its pharmacological and toxic effects. The second difference relates to its kinetics. Fentanyl has a rapid onset and short duration of action as compared to the commonly used opioid analgesics. With regard to the pharmacology of fentanyl-related substances that have been recently encountered by law enforcement, there is limited or no information. DEA has obtained preliminary pharmacology data on over 35 fentanyl-related substances through interagency agreement with other federal agencies and through research contracts. These data showed that these fentanyl analogs, similar to fentanyl, act as potent mu opioid receptor agonists. Thus, these novel fentanyl analogs are likely to have a toxicity and abuse potential similar to fentanyl. The two main pharmacological effects of opioid analgesics play a central role in their adverse impact on the public health. First, opioid analgesics are highly addictive class of drugs that activate reward pathways in the brain to produce intense euphoria. And second, in high doses, opioid analgesics consistently depress respiratory center in the brain. Deaths resulting from overdoses with these substances are most often due to respiratory depression, leading to complete failure of breathing. Fentanyl or its analogs, similar to other opioid analgesics, carry high risk of causing serious health consequences, including deaths. Because illicitly produced fentanyl and its analogs are manufactured in non-pharmaceutical settings under non-controlled conditions using crude methods, the purity of the final product is uncertain and inconsistent. Users of these products may end up consuming unknown amount of active drug or drugs and are at a high risk of adverse health outcome. Recent CDC data demonstrated that intravenous injection is the most common route of administration in deaths related to fentanyl and its analogs. Because intravenous route of administration puts these drugs directly into the bloodstream, it speeds up the accessibility of these drugs to their site of action in the brain, thus leading to enhanced effects and toxicity. The intravenous route of administration combined with high potencies of fentanyl and its analogs and distribution of products with the unknown composition of drugs and their amounts collectively put individuals who consume these products at even higher risk of overdose and overdose deaths. Thank you for this opportunity to briefly discuss pharmacology, toxicity, adverse impact on public health related to these substances. I'll be happy to answer any questions you may have. Thank you.
Thank you, Dr. Tella. Dr. Logan?
Chair Pryor, Commissioners, again, thank you for the opportunity to address the Commission this morning. As was mentioned in my introduction, I represent a laboratory that performs toxicology and sees drug testing on behalf of law enforcement agencies and coroners and medical examiners' offices. We have a pretty broad perspective on the extent of the impact of the opioid epidemic by virtue of the fact we tested over 60,000 death investigation cases for coroners and medical examiners last year, and examined over 68,000 exhibits, seized drug exhibits from law enforcement agencies, some in support of backlog elimination projects, as was referenced in one of the earlier questions. Based on our experience, the continued increase in rates of positivity for fentanyls in our casework has continued this year. In the 18 months leading up to September of 2017, we had over 17,000 death investigation cases that were positive for fentanyl from testing coroner and medical examiner casework. That was approximately double the rate in the prior 18 months. And our data from this year indicates that the positivity for the year will likely be 30% in 2017 over what it was in 2016. The impact of the increase in opioid availability is not limited, however, just to fatalities. In our drug-impaired driving casework and vehicular homicide casework, it took 4 years from 2011 for fentanyl positivity in these cases to increase by a factor of 3. However, the rate of positivity increased threefold again in 2016 alone, and has increased by a further 30% in 2017. In total, about 14% of our drug-involved DUI cases now are positive for fentanyl. In terms of challenges for laboratories, the number of compounds in the fentanyl category that our laboratory tests for has increased dramatically, putting additional demands on resources in the laboratory, and that's also the experience of laboratories in the public sector. We were testing for 4 fentanyl analogues in 2011, 7 in 2014, 22 in 2016, and this year over 35 different unique chemical compounds are part of our testing menu. When you consider other non-fentanyl opioids, including, as was mentioned, U-47-700 and some analogues of that substance which are now appearing, we're testing for over 40 new opioids over and above the traditional morphine, heroin, and oxycodone drugs. That puts a significant burden on laboratories. We, as a large laboratory, have more resources for research and development, but many of the smaller laboratories at the state and local level do struggle in keeping up with the changing menu of substances, and they do rely on laboratories like ours as a resource for assistance. The second issue I wanted to address was about the impact of some of the unknowns regarding the toxicity of these substances. As forensic toxicologists, myself and my staff are called to go to court to testify about the significance of toxicology findings in death investigation cases. And frequently the question that's asked is, but for the presence of a particular opioid, would the decedent have lived or died? When new substances new drugs are created by pharmaceutical companies, they go through an extensive testing process. It starts with in vitro studies in the laboratory, progressing to animal studies, and then to careful human trials, and then subsequently to post-launch monitoring of adverse effects of these substances. None of these safeguards or screenings are done for these illicit substances. So a lot of the information we have about drug toxicity for pharmaceutical drugs, we simply don't have that for some of these novel, illicit, clandestine drugs like fentanyl and its analogues. So that does limit the extent to which that question can be reliably answered, and I know that that is frequently a frustration for investigators and in the prosecution of drug delivery, homicide, and drug intoxication cases. There are some positive changes that have taken place that support the laboratories that are responding to this crisis, including availability of new technology with greater sensitivity and better discriminating power. Those technologies tend to be quite expensive, and many laboratories don't have access to them. But we have a better understanding of the mechanisms and methods for identifying new substances as they appear, and I think we're doing a better job of keeping up now than we were a couple of years ago. Thank you.
So, Dr. Logan, I understand from your written submission that you think that fentanyl analogues do constitute a class that are subject to course structure scheduling. Is that right?
Yes.
Why is that?
When you look at the chemical composition of the substances, they have three characteristic domains, and those are shared to some extent in everything that would be considered to be part of that class. So minor modifications to those domains do impact the potency of the drug, but if you identify the presence of these chemical substituents on the molecule, if all three of them are present, then a chemist can recognize them as being related to or derived from fentanyl.
Thank you.
I have one question. Dealing with potency of the analogs, are you able to state with any certainty whether the vast majority of the analogs that you're seeing are significantly more potent than fentanyl itself?
I can address that. We recently tested 7 fentanyl analogs. They were slightly less potent than fentanyl, but more potent than morphine. But some of the fentanyl analogs we encountered in late 1970s and 80s, they were almost close to fentanyl in terms of the potency. But the recent ones, so far we tested in animal studies, 7 of them showed slightly less potent than fentanyl, but more potent than morphine. But we are still in the process of testing 20 more fentanyl analogs. We don't have the data, but in vitro data, we have about over 35 substances. All of them are close to, not actually close, but some of them are less, some of them are more potent, but a lot of them are close to fentanyl. But this is, again, in vitro data, so we really can't extrapolate exactly what it is going to do once we inject into the system body in animals or humans. So, but based on the in vitro data, it looks like a lot of them are potent.
Dr. Logan, would you agree with that, or are you seeing things different in the laboratories?
From reading the literature, I would agree with that assessment. We recently did a review of a number of the patents from the 1970s and 1980s where pharmaceutical companies were developing and experimenting with a variety of different fentanyl analogues. There are over 600 fentanyl-related compounds in those early patents. A small percentage of them do have animal data that support quite a range in terms of potency, from many hundred times more potent in animal models for analgesia than fentanyl to compounds that are less potent. And in some cases, if the molecule is modified sufficiently, very little potency.
Judge Breyer, do you have any questions?
Yes, I do. Dr. Logan, I have two questions. Maybe the first one may be fairly obvious. Does fentanyl in and of itself have addictive characteristics or qualities? That's one question. The other question is there any scientific evidence as to the quantity of fentanyl in terms of being mixed with heroin that would be a safe dose or would be a lethal dose? Is it simply a trace of fentanyl that is used in heroin that would cause a death, or does it have to be more than that? You could ask this question.
So to address the first question, the fentanyl acts on the same receptors in the brain and the central nervous system that morphine or heroin or other opioids act on. So it does produce the same constellation of effects, and it does produce the same potential for becoming addicted or habituated to the drug.
Okay.
With respect to the effect of combinations of the drug, I think our experience is that the composition of drug mixtures is extremely variable. Sometimes the fentanyl is a trace in the heroin product, sometimes it's the inverse. But for drugs that act on the same receptors as heroin, fentanyl analogues do, if they're all contained in the same purchased dose of an opioid, their effects will be at least additive, if not synergistic, meaning multiplicative rather than additive.
Well, let me follow up on that. What I'm trying to figure out is whether we have to be concerned that if some quantity of heroin has a trace of fentanyl in it, that would render it either potentially lethal or certainly lethal. Or is there a certain percentage of fentanyl in a heroin combined with heroin that one could say, well, that the sale of that kilo of heroin with X amount of fentanyl in it would certainly constitute a lethal dose? And I understand there are a lot of variables, cut and so forth and so on, but I'm just trying to figure out to what extent, since fentanyl seems to be far more lethal than heroin, as I understand it, potentially lethal. I'm trying to figure out in my mind, is there some quantity of fentanyl in a heroin compound or mixture that would increase the risk, substantially increase the risk of death?
So I would be reluctant to try to put any quantitative number to that in terms of what the incremental risk is from the addition of fentanyl to heroin. Both are inherently dangerous. The users typically have no idea of what the dose is when they're taking the product. They certainly wouldn't know the relative amounts of the drugs in the substance that they've purchased.
Would not?
Would not.
Right.
So I think it's difficult to say, is there a threshold amount of fentanyl that you could add to heroin that would not make it more dangerous? I mean, there is, but what that number is, I think, is not known.
Well, let me follow up on that. In other words, as it is now, we punish people who furnish drugs based upon, one, the nature of the drug, and two, the quantity that's sold. So you take that now. Let's look at it in terms of fentanyl. And is there any way you could say this quantity of heroin, because it has fentanyl in it, is twice, 5 times, 10 times more dangerous than simply the heroin uncut or unadulterated by fentanyl? Do we have some evidence on that issue, or is that simply still unknown?
Well, for fentanyl itself, since it was developed as a pharmaceutical, there are data that indicate what the relative potencies of fentanyl and are to morphine or to heroin. So you could calculate the relative potency of mixtures with different proportions of heroin and fentanyl and compare that back to the potency of unadulterated heroin. However, for the vast majority of the analogs, there is no such equivalency or data that would allow you to do that kind of calculation.
Thank you.
Just one follow-up. Of course, at the street level, you're not getting an exact mixture anyway, are you? You may have 1 gram of fentanyl mixed with a kilogram, one portion of the kilogram, and one user may get all of that. I mean, it's not a...
Yeah, there's very little quality control.
The other follow-up question I had for you was I think you referred to as the synergistic effect. And I'm interested in drug users that are not using the same type of substance such as heroin, the opioids, but methamphetamine that has different effects on the...
Yes.
Have you observed that, and have you been able to testify about that? Or?
We do see many polydrug cases, both in death investigation casework and impaired driving casework. The constellation of effects is a mixture. Certainly, if you take a stimulant and a depressant, they don't cancel each other out. You tend to see the worst of both drug classes in terms of their adverse effects.
Okay, thank you, Dr. Van Lin, Dr. Tella, and Dr. Logan. We appreciate your oral presentation this morning. Of course, we have your more extensive written testimony as well, and thank you for being here.
Thank you.